Hello everyone, welcome back to Science with.
Noor and Sakina.
In our last video, we outlined the importance of energy in muscle contraction, protein synthesis, DNA replication, cell movement, and etc. We also explained what is ATP as an energy currency molecule. In this video, we are going to explain how ATP is synthesized from the first two stages of aerobic respiration, glycolysis and link reaction. To begin with, respiration is a chemical reaction that happens in all living cells, as it is the way that energy is released from glucose, so that other chemical processes needed for life can occur. Please don't confuse respiration with breathing, which is called ventilation. To get this out of the way, the, the biological process of inhaling and exhaling gases between cells and their surroundings is referred to as breathing. Breathing involves several respiratory structures, including the windpipe, lungs, and nose. In contrast, respiration is a chemical process which occurs in the cell. Now, let's move on to the definition of glycolysis. Glycolysis is a cytoplasmic pathway, which means it takes place within the cytoplasm, which breaks down glucose into three carbon compounds called pyruvates and generates energy. An overview of the process. Glucose is the process by which Glycolysis is the process by which glucose is converted into two pyruvate molecules, resulting in the production of two molecules of ATP and two molecules of NADH. Glycolysis is an anaerobic reaction and, in low oxygen conditions, is the sole source of ATP. Glycolysis can be considered as a two-part process. First is energy investment phase, which requires two ATP molecules to produce high-energy intermediates. Second is the energy payout phase, in which the intermediate is metabolized, producing four ATP molecules and two NADH molecules. Now, let's move on to glycolysis reaction steps. The first step is the conversion of glucose into glucose 6-phosphate. The enzyme that catalyzes this reaction is hexokinase. The second step is the rearrangement of glucose 6-phosphate into fructose 6-phosphate by glucose phosphate isomerase. In the third step, phosphofructokinase with magnesium as a cofactor changes fructose 6-phosphate into fructose 1,6-bisphosphate. The fourth step is the enzyme aldolase splits fructose 1,6-bisphosphate into two sugars that are isomers of each other. These two sugars are dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. The fifth step, the enzyme triose phosphate isomerase rapidly interconverts the molecules DHAP and GAP. Glyceraldehyde phosphate is removed and used in the next step of glycolysis. GAP is a sole molecule that remains in the glycolytic pathway. As a result, the enzyme TIM acts on all the DHAP molecules produced, reorganizing the DHAP into GAP so that glycolysis can continue. We have two 3-carbon molecules at this point in the glycolytic pathway, but we have not fully covered glucose, but we have not fully converted glucose into pyruvate yet. In the sixth step, glyceraldehyde 3-phosphate dehydrogenases and phosphorylates glyceraldehyde 3-phosphate producing 1,3-bisphosphoglycerate. In the seventh step, phosphoglycerate kinase transfers a phosphate group from 1,3-bisphosphoglycerate to ADP to form ATP and 3-phosphoglycerate. The enzyme phosphoglyceromutase relocates the P or the phosphate group from 3-phosphoglycerate from the third carbon to the second carbon to form 2-phosphoglycerate. In this way, the enzyme is also restored to its original phosphorylated state. In the ninth step, the enzyme enolase removes a molecule of water from 2-phosphoglycerate to form phosphoenol pyruvic acid. In the last step, the phosphate group attached to the second carbon of PEP is transferred to a molecule of ADP, yielding ATP and forming pyruvic acid. The enzyme involved is pyruvate kinase. Since there are two molecules of PEP, we generate two ATP molecules. The net products of glycolysis at the end of the reaction are two pyruvate molecules, two ATP molecules, and two NADH. Although 4 ATP molecules are produced in steps 7 and 10, the net gain of glycolysis is only 2 ATP molecules because 2 ATP molecules are used in steps 1 and 3 of glycolysis.
Moving on to the link reaction. Here are some general points about the link reaction. It's called link reaction because it links glycolysis, where the oxidation of glucose begins, with the Krebs cycle, where the oxidation is completed. It occurs in the mitochondrial matrix. It occurs only in the presence of oxygen, so it can be considered as the first stage of aerobic respiration. In this link reaction, no ATP is produced. Link reaction steps. In the first step, pyruvate is transported from the cytosol into the mitochondrial matrix by carrier proteins on the mitochondrial membrane. The transport process is called active transport. In the second step, the pyruvate loses a carbon atom by decarboxylation, which forms a carbon dioxide molecule. In the third step, the two-carbon compound then forms an acetyl group when it loses hydrogen atoms via oxidation. NAD is reduced to NADH. Pyruvate undergoes oxidative decarboxylation, and the enzyme complex involved in the reaction is the pyruvate dehydrogenase complex. In the fourth step, the acetyl compound then combines with coenzyme A to form acetyl coenzyme A, or acetyl-CoA. Fatty acids from fat metabolism may also be used to produce acetyl coenzyme A. Fatty acids are broken down in the mitochondrion in a cycle of reactions in which each turn of the cycle shortens the fatty acid chain by a 2-carbon acetyl unit. Each of these can react with CoA to produce acetyl coenzyme A. acetyl coenzyme A, which like that produced from pyruvate now enters the Krebs cycle. Moving on to the products of the link reaction per glucose molecule. 2-acetyl coenzyme A, 2 reduced NAD, and 2 carbon dioxide molecules. As glycolysis splits glucose into 2 pyruvate molecules, the link reaction occurs twice per molecule of glucose. In the next video, we will explain the Krebs cycle and oxidative phosphorylation. Thank you for watching.